This is an overview of an article titled A Prospective Study of Epigenetic Age Acceleration and Incidence of Cardiovascular Disease Outcomes in the Atherosclerosis Risk and Community Study, published in the March 2018 issue of Circulation, Genomic and Precision Medicine. DNA methylation is a widely studied epigenetic process by which methyl groups are added or removed from the DNA sequence, usually at cytosine guanine dinucleotides, or CPGs, impacted by genetic and environmental stimuli. Levels of DNA methylation at specific locations around the genome can be used to assess biological aging and to determine a measure called epigenetic age acceleration, which is calculated as the difference between an individual's DNA methylation predicted age and their actual chronological age. A positive epigenetic age acceleration measure is known to be associated with some risk factors for poor health and all-cause mortality. However, little is known about whether epigenetic age acceleration is related to the development of atherosclerosis, or cardiovascular disease. In the paper under discussion, the authors calculated two epigenetic age acceleration measures, one devised by Horvath and one by Hannum and colleagues, using whole blood samples from 2,543 African Americans from the general population in the Atherosclerosis Risk and Community Study, or ARIC. Linear regression and Cox regression, respectively, were used to assess the association of epigenetic age acceleration with carotid intima media thickness, or carotid IMT, and incident cardiovascular events, including cardiovascular disease mortality, myocardial infarction, fatal coronary heart disease, peripheral arterial disease, and heart failure, all over a median 21 years of follow-up. The Horvath and Hannum clocks are epigenetic scores that assess biological age from DNA methylation patterns. Both algorithms were developed in large samples covering most of the lifespan and various ethnicities. Hannum and colleagues developed a model based on 71 CPG sites using DNA from whole blood samples, that is, blood-specific, while Horvath developed a similar model based on 353 CPG sites using DNA from samples of 51 healthy tissue and cell types, making it pan-specific. The Horvath clock can be used to compare the ages of different tissues, cells, and organs from the same individual. In the present study, the authors first presented consistent associations among the two versions of epigenetic age acceleration and numerous cardiovascular disease risk factors. For example, age acceleration was on average 0.7 to 1.5 years lower in women compared to men, 1.7 years higher in current smokers compared to never smokers, and 1 year higher in those with diabetes compared to those without diabetes. In multivariable models, additionally including C-reactive protein, which is a measure of inflammation, an estimated glomerular filtration rate, which is a metric of kidney function, CRP was positively associated and eGFR was negatively associated with the Horvath and Hannum measures of epigenetic age acceleration. This figure shows that both the epigenetic age acceleration measures and chronological age were positively associated with carotid IMT. Specifically, each 5-year increment in Horvath or Hannum age acceleration was associated with higher carotid IMT upon minimal adjustment for chronological age, sex, and estimated leukocyte cell type abundances. These associations persisted after further adjusting for education, body mass index, health behaviors, and clinical risk factors. The authors also evaluated the association of epigenetic age acceleration with incident cardiovascular disease. In this figure, we can see the log hazard ratio of fatal coronary heart disease, or CHD, increasing across the distribution of Horvath epigenetic age acceleration in a minimally adjusted model. The Hannum age acceleration measure showed a similar and slightly stronger association with fatal CHD. Similarly, this figure illustrates the log hazard ratios of peripheral arterial disease, or PAD, as well as early incidence of heart failure, also increasing across the distribution of Hannum and Horvath epigenetic age acceleration measures in minimally adjusted models. Age acceleration was nominally associated with PAD and heart failure independently of chronological age and other traditional cardiovascular disease risk factors. Chronological age was a robust marker for future risk of cardiovascular disease mortality, myocardial infarction, fatal CHD, PAD, and heart failure. Comparatively, the epigenetic age acceleration measures were only modestly associated with most of the cardiovascular outcomes. Associations with incident cardiovascular outcomes were generally consistent between the two versions of epigenetic age acceleration, being slightly stronger using the Hannum measure. This is in spite of the Horvath and Hannum measures using largely non-overlapping sets of methylation sites in the genome. In an attempt to focus on specific pathophysiological mechanisms, and possibly identify potential future targets for intervention, the authors also looked at specific CPG sites to see whether any individual probes used to estimate age acceleration are by themselves markers of cardiovascular disease risk. DNA methylation in one site in particular, near the galanin receptor 1 gene on chromosome 18, was significantly associated with the risk of fatal coronary heart disease after taking multiple testing into account, and additionally was associated with all other cardiovascular outcomes at nominal significance. However, as the authors clearly stated, this association needs to be replicated in an independent study. In conclusion, in this sample of African Americans, increased epigenetic age acceleration in whole blood was a risk marker for incident fatal CHD, PAD, and heart failure independently of chronological age and traditional cardiovascular disease risk factors. DNA methylation-based measures of biological aging may help to identify new pathophysiological mechanisms contributing to the development of CVD. Future studies should investigate smoking and diabetes as potential modulators of epigenetic age acceleration.